Hi, good morning, everyone. Today we will discuss about the root, one of the important part of the plant, So if we see from the function of the root, there are several important function in plants, The first one, they anchor the plant in the substratum, so for supporting them, supporting the plant. Second one, the principal organ for water and mineral uptake from the surrounding environment. So the plant needs them, for growing, of course, and also for the photosynthesis, and the plant cannot produce themselves, so they need it from the surrounding, and in some plants the root also has a function for the storage of food reserves. Maybe you still remember regarding the function of phloem, that can transport the result of photosynthesis through all of the plant part. If the root produced in the soil by a plant, collectively we call it as a root system. The root system begins its development from the embryonic radicle that grows out of the seed and from the radially symmetrical primary root. And the secondary lateral loop develop from the primary root, and each of these will turn from new lateral roots of tertiary rank. So so if we see, there are two morphological types of root system. The first one is fibrous, and the second one is taproot system. So what is fibrous root? The fibrous root composed of large numbers of roots that are nearly equal in size. The root system of this type are found in the grass. A taproot is the primary root remains the largest root, and a number of smaller roots are from it. All monocot roots are adventitious because they arise from a stem or a leaf or other part of a root. And then the apex of the root is covered by a mass of loosely organized cell. We call it the root cap. In addition to serving a protective function, the root cap is active in mucilage production and as gravity sensing structure. Just behind the root cap is the meristematic area in which cells from an elongation follows. So if you see this, I hope that you can imagine what are the differences between the taproots and also the fibrous roots. So if you take a look at the slide, for the taproots, we can find it in dicots. Usually it is long and then thick root, hickory, for example, hickory and oak trees. While for the fibrous roots, we can find it in monocots. So if you see this fibrous root, we cannot see the larger part of the root, which is larger than the other. So And if you see from the slide, we can see that the branch is very extensive. Yeah, a lot of branch. And if you see, the branch is has many thin roots. Yeah. Yeah. So this is the fibrous root system. Yeah. Like a tangled mass of roots. And they are some example of the plants like banana, coconut, rice, corn, and also sugarcane. If you see based on the classification, yeah, I think some of the plants here are classified as grasses, yeah. And then if you see this, yeah, for the tap roots, it has large main central root, yeah. And then they also have many branching roots, and usually it can go deep in the soil, yeah. So for example of the tap roots are large trees, yeah, like a mango, avocado, yeah. While for the other example for shrubs, yeah, like tomato and also peppers, yeah. So now we will discuss about the structure of the root, yeah. Mainly, yeah, the structure of the root can be divided as epidermis, cortex, endodermis, pericycle, and also the vascular system. Yeah, so this is the I think the two-dimensional for the root here. Yeah, so you can see here the epidermis, yeah, and then the ground tissue for the cortex, and then there is a zone of maturation, and then zone of elongation, and the lowest part of the root, it is root cap. Yeah. So here, yeah, for the epidermis, yeah, in all root, in some part of the plant, yeah, root hair are usually present, yeah, as a short distance from the growing apex, yeah. This develop from the rhizodermis or root epidermis, yeah, and often the root hairs arise centrally from the basal part of the cell, and occasionally they arise from near one end, and because of this feature, yeah, it is useful for diagnostic, yeah. And then for a short distance further away from the apex, that the root hairs often die and shrivel, but in some plants the root hair persists for a long time, yeah. And then to the inner side of the rhizodermis, an exodermis may develop, yeah, particularly in the monocots, yeah. And then this characteristically consists of angular cell with some thickened and also lignified walls. And then a multiple epidermis or filament is found. And then for the example, in the aerial roots, it is epiphytes, yeah, and also aeroids. And then frequently cells in situation have specialized spiral or reticulate or irregular thickening bands, and are capable of storing water absorbed from humid atmosphere, mist, or rain. Yeah. So if you see that this is the root epidermis, Yeah. The outermost, it is a single layer of cells that have a function. Yeah, the first one to protect, yeah, especially from the disease, and also for absorbing, yeah, the water and nutrients from surrounding. And then if you see the root here, yeah, here, the root here is a tubular extension of the epidermal cell. And the function, one of the function of the root hair is to increase surface area of root for better water and nutrient absorption. So with the help of the root hair, yeah, the absorption for, especially for water and also for mineral, it will be better. It will become better. Yeah. Nah, this is the figure, yeah, for the root hairs where the water and mineral were absorbed. Yeah. So this is, yeah, so if you see in the bottom part of the figure, you can see root hairs. Yeah, here the root hairs can increase the surface, yeah, for the better absorption. Okay, and then the next one is cortex. Yeah, there are two basic types of cortex, and they are recognizable among other less frequent variation, and these are the solid and the lacunate cortex. Yeah, the solid one is composed of the parenchymatous cell, which are relatively compact, yeah, compact, with the intercellular spaces confined to the angle between cells. It is usual for there to be a gradual increase in the size of such cells from the outer to the inner layers. But the innermost few layers are frequently of a smaller and more compact type of cells. Yeah, and such an arrangement is common in both monocotyledon and dicotyledon on plants which grow in well-drained soil types. Yeah, so here it is the figure of the cortex. Yeah, you can see usually this kind of figure, yeah, in the materia medica, yeah, and also in so this is the, so if you see here, yeah, this is the result of the SEM, yeah, scanning electron microscope. Yeah, so there is a 75 zoom, yeah, high, bigger than the real structure. Yeah, and then the second type of cortex, yeah, it is lacunate cortex, which has a few outer layers and compactly arranged cells, and the innermost layers may be similarly compact. Yeah, and between this, the radiating plates of cells are in. Yeah, with a large air space between them. Yeah. And if you see the diaphragm of stellate, yeah, literally, what is stellate? It is a star-like cell with radiating thin arm-like protrusion. Yeah, and other cell types may traverse, yeah, the lacunate and as in the stem. And most plants with this type of root cortex have their roots in soil, which is periodically waterlogged or even immersed in water. Yeah. So the condition of this, we can find it in Zea, yeah, which normally lacks lacunae by growing the plants in the waterlogged soil. Scleroid, yeah, fiber, tannin cell, mucilagous cell, and also crystal containing cell can be found scattered in the parenchymatous. Yeah, yeah, I think it is a wide range of families, so not only special cases. Yeah, so yes, sometimes, yeah, based on the like a crystal or a scleroid or fibers, yeah, we can also make them as a marker fragment, yeah, for the fragment. Yeah, like in the next statement, yeah, the presence and also the distribution can be helpful in narrowing, yeah, narrowing down possibilities for the identification, but are rarely of taxonomy significance. Yeah, so nowadays, yeah, the development of identification for the plant, yeah, it is related also with the PCR, yeah, and so you will know the sequence of the DNA, yeah, and you will know also, yeah, it is for the identification of the plant. But I think if you use the PCR, for example, I think we have to pay more, yeah. I think it is I think more economically if you can make the identification of the plant, yeah, based on the microscopic and macroscopic, yeah, of the plant. So here, yeah, so the root ground tissue, yeah, like a cortex, yeah, it provides support and often stores sugars and starch, yeah. And then this is for the root cortex, yeah, especially for the Casparian strip, yeah. So here there is an explanation for the Casparian strip, yeah. It is a water impermeable strip of waxy material, yeah, found in the endodermis, yeah. So what is endodermis? It is the innermost layer of the cortex. And then for the Casparian strip, yeah, it will help to control, yeah, to the uptake of the mineral into the xylem, yeah. So I hope you still remember the difference between xylem and phloem, yeah, and also the process when the water and mineral, yeah, were taken, yeah, by the xylem. Here, yeah, some of the figure, yeah, especially for the endodermis, yeah, it is also, yeah, the figure of the endodermis also can be a fragment, yeah, can be a fragment for identification of the plant, yeah. And then this is the explanation for the endodermis, yeah, the cortex on its inner side abuts onto the endodermis, yeah. This characteristic physiologically active tissue is frequently one layer thick, but in some plants it can be two or more layered, yeah. And then although the endodermis can be composed of the cell with evenly thickened walls, in the majority of the plants, yeah, the inner and anticlinal walls are more heavily thickened, yeah, with the lignin and also suberin than the outer periclinical walls, yeah. Consequently, in transverse section, They are readily distinguished from adjacent cell layers, and so we call as a U-shaped thickening. Yeah. And then at the intervals, yeah, the certain cell of the endodermis are thin-walled. These are the passage cells and are usually the opposite of protoxylon poles. Yeah, and then they are supposed to afford a more ready pathway, yeah, from root hairs via the cortex to the protoxylon elements, especially for water and dissolved solutes. Yeah, and the other cells in the endodermis are supposed to restrict water, yeah, restrict water flow, yeah, between cortex and also the stellule. Yeah, and then for the anticlinal walls of all endodermal cells are equipped, yeah, with the spatial suberized waterproof impregnation, and also the Casparian strip or bands with the cell membrane, the plasma lemma attached to the Casparian strip. Yeah, and then the next part is pericycle. Yeah, what is a pericycle? Yeah, the cells of the next layer, yeah, to the inside, and usually narrower than those of the endodermis. Yeah, and they frequently have relatively thin walls. Yeah, and then roots lack any nodes, and lateral roots arise endogenously. There is their growing point or apices, yeah, first develop into the pericycle. And then the division of cell in this region produce a lateral root which has to grow, yeah, through the tissue of the endodermis and cortex to reach the exterior of the primary root. Yeah, and because of the pericycle bounds the vascular system of the root, and the vascular continuity between the new lateral root and the main root can soon be established once active growth has started. And then some roots may have quiescent. What is quiescent? The potential laterals in the pericycle, which require some hormonal stimulation, yeah, or the removal of a hormonal restraint before they develop. Okay, this is the last part, yeah, of the root. It is a vascular system. In most dicotyledons, there are between 2 and up to about 6 protoxylem strands alternating with phloem strands. And then the large number of species have triarch, yeah, 3-stranded, or tetraarch, yeah, 4-stranded arrangement, or a mixture of both. Yeah, if about 6 strands are present, the roots are described as polyarch. Most monocotyledon roots are polyarch. And then the metaxillem tracheids or vessel elements are normally conspicuous and are on the same radial axis as the protoxylem poles and to their inner side. So this arrangement is the characteristic of the roots and can distinguish them from the stems. And then root anatomy is initially exarch with the protoxylem poles to the outer side and in arch in stems, with the protoxylem to the inner side of metaxylem. Yeah, and then the transition usually occurs in the hypocotyl or the top of the primary root. Yeah, and then currently in monocotyledons there is one ring only, but there may be more or scattered metaxylem elements in the root center. Yeah, and in most dicotyledons, several metaxylem elements are grouped together in strands. Yeah, and then in the monocotyledons, the vessel element is thought to have had its origin in roots. Yeah, And then in the least advanced plants, if vessels are developed at all, they are found in the roots only and not in the stem or leaf. Yeah. And then for the next stage of advancements, yeah, for the vessel to occur in the root and the stem, and in most advanced plants, vessel occurs in the root, the stem, and also the leaf. And then a number of plants have shorter, wider vessel elements in the root than in the stem or leaf, yeah, bearing out the theory of the evolutionary sequence from the primitive narrow, long elements to the more advanced, shorter, wider elements. Yeah, vessel and trihydrate wall pittings and thickenings are similar to those of the stem wood. And for the phloem cells of the root also have the same range of forms and found in also the stem. And for the center of the root may be made up entirely of xylem in dicotyledon or in monocotyledon, or some dicotyledons, especially at the base of the primary root. It may contain a ground tissue composed of parenchyma with thin or thickened walls. Yeah, so you can see the figure 4.5 until 4.7 in the next slides, and also the scleroid could be present. Yeah, so here, yeah, this is the ranunculus. Yeah, we call it buttercup root. Yeah. Illustrating the relatively simple structure of young dicotyledonous root. The xylem is tetrarch and 4 strands of phloem alternate with the protoxylem. This root is just beginning to undergo limited secondary growth. Yeah. While here, yeah, this is the iris root and showing a very prominent endodermis. Yeah. Seen here as the layer of the cell with the streaking and thickening of the radial and inner tangential walls. Yeah, and the endodermis is the innermost, yeah, innermost of the cortex, and the wall thickening forces water and other molecules to take the symplasmic route, yeah, from the cortex to the stela or vice versa. So this is the also the portion of zeamice, yeah, or corn, yeah, using the arrangement, yeah, the separation of the cortical from the stellar tissue, yeah, using this the SEM, yeah. So you can see the smaller part of the plant. Yeah. Okay, so this is the end of the root, yeah, the root anatomy. So see you again for the next part of the discussion. Okay, thank you.